Welcome to the lecture series on control of gene expression. I'm Janine Post of the Developmental Bioengineering Group at the University of Twente. In this lecture, I will discuss regulation of gene transcription. Illustrations in this video are based on the book Essential Cell Biology, Chapter 7 and 8. In this second lecture, I discuss how gene transcription is regulated. Gene expression is regulated by transcriptional regulators that directly interact with the DNA using their DNA binding motif. The side chains of the amino acids in the DNA binding motif interact with bases in the DNA via hydrogen bonds with bases in the major groove, as is shown in this figure for the DNA binding domain called a homeodomain. Many DNA binding proteins contain such a homeodomain. In literature, you will often find the term transcription factor. The terms transcription regulator and transcription factor are mostly interchangeable, but transcription factors have to directly bind DNA and contain at least one DNA binding motif. Various motifs exist in addition to homeodomains, for example zinc finger, helix turn helix, and leucine zippers. Humans make up around 2,000 unique transcription factors, indicating the importance and complexity of these proteins in development and maintenance of a complex organism. Transcription factors often work together with other proteins in regulating transcription. These proteins include coactivators, chromatin remodelers, histone acetyltransferases, histone deacetylases, kinases, and methylases, which are also essential to gene regulation but lack DNA binding domains and therefore cannot be called transcription factors. Signals in the nucleotide sequence of a gene tell bacterial RNA polymerases where to start and stop transcription. Bacterial RNA polymerases, shown in light blue, contain a subunit called the sigma factor in yellow, that recognizes the promoter of a gene, shown in green. Once transcription has begun, the sigma factor is released and polymerases move forward and continue to synthesize the RNA. Elongation continues until the polymerase encounters a sequence in the gene called the terminator, shown in red. After transcribing this sequence into RNA, which is shown in dark blue, the enzyme halts and releases both the DNA template and the newly made RNA transcript. Polymerase then reassociates with a free sigma factor and searches for another promoter to begin the process again. Transcription factors bind at the start of eukaryotic genes. These factors are designated TF2B, TF2D, and so on. Most eukaryotic promoters contain a DNA sequence called the theta box. The theta box is recognized by a subunit of the general transcription factor TF2D, called the theta binding protein, or TBP. The theta box is a DNA sequence with a specific organization. The binding of TF2D enables the adjacent binding of TF2B. The rest of the general transcription factors, as well as the RNA polymerase itself, then assemble at the promoter. At this site, TF2H phosphorylates RNA polymerase II. This releases the polymerase from most of the general transcription factors, so it can begin transcription. The phosphorylated RNA pol II contains capping, splicing, and polyadenylation factors. Processing of the RNA happens immediately during transcription. All eukaryotic mRNA or messenger RNA molecules are modified by capping and polyadenylation. A eukaryotic mRNA has a cap at the 5' end and a poly A tail at the 3' end. In addition to the nucleotide sequences that code for protein, most mRNAs does also contain non-coding sequences. The non-coding portion at the 5' end is called the 5' untranslated region, or 5' UTR, and at the 3' end is called the 3' UTR. Capping and poly A tails mark the molecule as mRNA and increase its stability. Capping and poly-A tails also function as checkpoints before translation because the machinery knows that the molecule is complete. The 5' cap is also important for nuclear export as it is recognized by nuclear export proteins. It prevents mRNA from being broken down, and it helps the ribosome to attach to the mRNA. While bacterial genes are a continuous stretch of sequences encoding the amino acid sequence of a protein, the protein coding sequences of most eukaryotic genes, or exons, are interrupted by non-coding sequences, introns. The promoter sequences are indicated in green. By exon skipping, in which alternative exons are included or excluded in the mature mRNA, which is also called alternative splicing, creates different protein products from a single gene. The choice of which proteins are included is determined by the expression of various small nuclear ribonuclear proteins, SNRPs, which direct the cleavage of the RNA at the intron-exon borders and catalyze the covalent linkage of exon sequences. Different cells can express different of these SNRPs, ensuring that there are variants in the genes created in that cell type. For more information on this, see Chapter 7, Figures 720 to 723. A specialized set of RNA binding proteins signals that a completed mRNA is ready for export in the cytosol. As indicated on the left, the 5' cap and poly-A tail of a mature mRNA molecule are marked by proteins that recognize these modifications. Successful splices are marked by exon junction complexes. See figure 722. Once an mRNA is deemed export ready, a nuclear transport receptor associates with the mRNA and guides it through the nuclear pore. In the cytosol, the mRNA can shed some of these proteins and bind new ones, which, along with the poly-A binding protein, act as initiation factors for protein synthesis. So gene expression is controlled at many steps. These are the steps regulated by housekeeping proteins, such as general transcription factors. In the next part, we are going into more detail on how cells regulate which genes are expressed and which are not. Thank you for watching. If you have any questions, please email me. In the next lecture, we will be talking about how specialized cell types are generated.